G'day, my name is Dr. Budamananiakara and I'm a respiratory and sleep physician at Orange Health Service. I'm also honoured to be a lecturer in medicine at Charles Sturt University, as well as a clinical lecturer at University of Sydney. And it's with great pleasure that I welcome you to study medicine at Charles Sturt University. You guys must be very pleased with yourself, and you should be. It's a fabulous achievement and it's a remarkable honour that you get to study medicine and embark on this career. It's an amazing journey. There will be a bit of rough time, but there will be a lot of great highs, and it's really nice to welcome you guys to our club. So well done. Without much further ado, we'll go on to the lecture, which today is on oxygen, hemoglobin and blood. Now this is the first physiology lecture, and I'll be presenting a lot more physiology when it comes to your respiratory and sleep medicine blocks. So I think physiology is a fascinating subject, and it is absolutely crucially important to the study of medicine. It's essentially the study of form and function, and underpins how the body works. I'm very passionate about physiology. I think it's very important. It's very vital for my field of medicine, and I still rely on the physiological principles that I learnt in medicine in my medical school years to try and tease out some of the difficult aspects that don't fit nicely into a textbook. So I hope that you will try and pay attention. I hope that you will try and learn concepts rather than memorize lists, as it really is the concepts that's going to get you through when you learn physiology. So without much ado, let's get on to the body of my presentation. I want to introduce a case first. So this is a 42-year-old male. He's a farmer that I met a couple of years ago when I was a registrar in a country hospital. He's nice, he's well-mannered, he's supported by his brother and sister and his young family. He presented to the emergency department where I was working overnight. He had cough of 3 days. He had rusty sputum. He complained of fevers, of chills, of drenching night sweats in the couple of days leading to his admission. When I saw him, he was breathless, and this was getting worse with time. He was a very sick man. He was drowsy when you examined him, and his blood pressure was very low. It was 60 on 40 millimeters of mercury. Now that is incredibly low. He had a respiratory rate of 35 breaths per minute, which is very fast, and you could tell that he was working very hard. He had loss of consciousness suddenly, and this triggered a medical emergency call. And at that time, his oxygen saturations were 78%. This was despite getting oxygen. It was incredibly low. And despite our best efforts, he was pronounced dead. Now, this was his chest x-ray. And I think even without knowing what a chest x-ray is, I hope you can imagine some of the abnormalities. I'll just get my pen out. The right lung here, you can see, is nicely opaque, whereas the left lung here is completely opacified. This is full of pus. Now, to try and figure out what's going on with this case, I want to introduce 6 key principles, and this will underpin the message of my lectures. It will be broken into 6 parts. Firstly, we're going to focus on oxygen, and we're going to understand that by oxygen, through its role in something called aerobic respiration, it is vital to normal human functioning. We're going to then introduce the cardiorespiratory system, and we're going to introduce a concept that it is through the respiratory system that oxygen comes in, It gets taken up from ambient air. It gets transported through the circulatory system, through the heart, into the body, eventually to the cells, where the oxygen is able to go in to the mitochondria to produce a molecule called ATP, which is the energy currency of the cell. We're then going to introduce the concept that oxygen is poorly soluble in blood, and it must be carried by hemoglobin to support the requirements of oxygen. We'll then talk about the structure of hemoglobin, its evolutionary adaptations in order to carry oxygen. We're going to go in depth in regards to the molecular physiology of hemoglobin, and I really want you guys to focus on this because I think this is a very examinable topic. Next, we'll say that hemoglobin is carried in red cells, and to do that, red blood cells must have unique adaptations and structural modifications, and we'll explore that in greater detail. We'll then focus on how delivery of oxygen is arranged. Firstly, that it depends on the hemoglobin concentration, which should make sense at this time because it's the primary oxygen transport protein. It relies on cardiac output, which should also make sense because that's how blood travels around the body. And also it should depend on the amount of oxygen that the molecule of hemoglobin can carry, or the oxygen saturation. So let's focus on the first concept, that oxygen, by virtue of its role in aerobic respiration, is vital to normal human functioning. Before we do that, let's focus a bit on history. In 1774, there was a very famous English scientist by the name of Joseph Priestley. Now, he was a scientist, he was also a priest and a theologist. And what he discovered was oxygen. So oxygen was first discovered in 1774. And the way it came about was that scientists had been studying what happens to gases for many years now. And what they used to do was they used to put a mouse in a beaker with a candle in it. And what they found was that a mouse very quickly died as soon as the candle flame was extinguished. And we now know that that is what occurs because the candle with the flame is essentially a form of combustion, and that converts oxygen to carbon dioxide and water, so depletes or devitalizes the air of oxygen. What scientists also found was that if you do this experiment in the presence of a plant, that the mouse can still survive. Now, the reason for this we now know is through the process of photosynthesis. A plant will produce oxygen, and this will sustain life. So what Joseph Priestley did was he had this compound here called mercuric oxide, so HgO2, and he didn't know the chemical composition at that time, but what he did was he focused light energy and essentially caused a burning or increased the heat of this. And what that did was it caused this chemical reaction. So it liberated the mercury, but it also caused oxygen to be liberated. And that oxygen obviously filled this air in this container, and that was able to sustain this candle flame 5 to 6 times longer than normal air would. So that air here, or that gas here, is enriched in a substance which was essentially the first demonstration of oxygen. So Joseph Priestley is first credited for finding out oxygen. Let's take another step and look at a cell. So by now you would have had a lecture on a normal cell, and you understand that cells have multiple components and it does multiple things. Let's take a nerve cell, for example, or a neuron. Now, neurons are important in the central nervous system for communication and for transmitting electrical signals. Now, that must require large amounts of energy. Let's consider the immune system, and let's imagine that this is a white blood cell. Now, a white blood cell will move and move and move until it captures a bacteria. It'll move and it'll envelope it in a process called phagocytosis, and it'll internalize it and eat it, essentially. That process of moving through using microtubules like here and actin filaments, that all must take energy. It doesn't come anywhere. And also cells must divide. And that process requires you to replicate the DNA in the nucleus, produce more proteins in the cytoplasm through ribosomes. That all requires energy. But where does this energy come from? What is the energy currency of the cell? Well, it turns out that the energy currency of the cell is a molecule called adenosine triphosphate. And by breaking adenosine triphosphate into adenosine diphosphate, releasing an inorganic phosphate releases a large amount of energy. And we can see why this is. So let's have a look at the chemical structure of ATP, or adenosine triphosphate. Here we have adenosine, which is a ribose sugar. And you can see each of these phosphate groups, or moieties as we call them in chemistry, are negatively charged. The phosphate atom is large, the oxygen atom is large, and you can imagine big, large, negatively charged anions cramped up in a small space in this molecule will cause this energy to be huge. It's like a ticking time bomb. And what happens is when you hydrolyze it, so when you remove one of these phosphates, suddenly instead of 3 negative charges, you get only 2 negative charges. So we go from 3 here and we go to 2 here. That causes a marked decrease in the energy of this molecule, and it's released, and it's this energy here that can be used to do cellular work. So how is ATP produced, and where does oxygen come in? Well, ATP is to generate ATP you require energy. Energy has to come from somewhere, and it turns out that it comes from breaking down large carbon-to-carbon bonds in molecules such as carbohydrates, fats, and proteins. Let's take a very simple example of glucose, which is the most abundant energy substrate that we have. You can see glucose has 6 carbon atoms. They're arranged in a ring, and each of these carbon-carbon bonds contains considerable amount of energy that can be unleashed to form ATP, and then ATP can be unleashed to do work. That's the philosophy, that's the logic. So how does this all occur? Well, we'll cover this in much greater depth through your nutrition block, but what happens is the glucose in the cellular cytoplasm gets broken down to pyruvate, which are 3 carbon molecules. So you can see there's 3 carbons here, and that process yields 2 molecules of ATP. Now this process that occurs in the cytoplasm is independent of oxygen. It is called anaerobic metabolism. Anaerobic metabolism. Now, if you had oxygen, what happens is these pyruvate molecules then enter into the mitochondria, where they undergo various chemical reactions, reorganizing of its molecular structure, liberating carbon dioxide as a waste product, and eventually, through the use of oxygen, ATP in sufficient quantities was generated. With anaerobic respiration, without oxygen, only 2 molecules of ATP are produced. When you add oxygen, you get an additional 34 molecules of ATP. If you only relied on anaerobic respiration, you would not have enough energy for normal cellular function to sustain life. That's why those mice died. They didn't have any energy to do any cellular work. The cells failed, therefore their organs failed, therefore the whole organism fails. Here, if you have oxygen, Remember, through the release of oxygen through mercuric oxide, that was able to sustain that candle. It was able to also sustain life. So oxygen through aerobic respiration generated sufficient quantities of ATP. Let's just go through a couple of metabolic concepts before we go further. Firstly, the Krebs cycle is a series of chemical reactions involved in reorganizing the two carbon bonds, and by doing this it transfers electrons into these compounds, NADH and FADH2. These are heavily electron-rich compounds, and by being electron-rich, they are full of energy and they're waiting to donate their electrons. And by donating electrons, they go into they release energy, and that energy is coupled to produce ATP. The final electrons have to go somewhere, and it turns out that this is where oxygen comes into. The electrons are accepted by oxygen. Oxygen gets reduced to water, and that's how oxygen participates in this transport cycle. Without oxygen, this process cannot occur. Let's take the electron transport chain and look at it in a bit more greater detail. You can see these high energy electron molecules, NADH and FADH2, that was generated through the Krebs cycle. They get oxidized, so they lose electrons, and by the process of losing electrons they generate energy, and that energy is used to pump these protons to create a strong electrochemical gradient, which can then be utilized by this pump called F-type ATP synthase to generate ATP. The process of putting one phosphate molecule into ADP, which is already having two negative charges, requires quite a bit of energy, and that energy comes from this pump, which is fueled by these protons, which are positively charged and concentrated within the inner and outer mitochondrial space, which is being generated by these transport proteins through coupling. The energy required to transport a proton up its electrochemical gradient with the energy released from oxidation of these substrates. The final electron carrier, of course, is oxygen, and oxygen accepts the electron and gets reduced and becomes water. Without oxygen, this process cannot occur, and if this process cannot occur, we do not have ATP produced through oxidative phosphorylation. So this process cannot occur. So without that's no oxygen. And remember, that is anaerobic respiration. And with anaerobic respiration, what tends to happen is you get pyruvate building up. Pyruvate cannot enter the cytoplasm, and when pyruvate builds up, to regenerate this molecule called NAD plus, we need to convert it to lactate. That is fundamental. If you do not have this step, you cannot generate NAD plus, and this system of glycolysis fails. So this is why pyruvate is converted to lactate. And in the process of no oxygen, this is the only way you can generate ATP in very insufficient quantities, but it's our only available source. And you can imagine that lactate builds up. So this is the basis behind lactic acidosis. So if you can imagine, when you're exercising or you're doing, you're pumping out a lot of weight, you can imagine that burn and that sting in your muscles. That's because the amount of oxygen is insufficient to supply the amount of ATP that's required to do work. So you rely also on anaerobic metabolism, and that by necessity generates large quantities of lactate, which is an acid, which causes that burn. Now, you can also imagine that if you don't have oxygen, that's a bad situation. So lactate can also be used as a biomarker for poor tissue perfusion. Now, there are other reasons for a raised lactate, but suffice to say, it can also be a very bad prognostic marker. And let's take this study, for example. So in this study, what they did was, I think this study was based in a Scandinavian country, and what they looked at was all patients that presented sick to an emergency department in one of the Scandinavian countries. And what they did was they took the lactate levels of all the people, and then they stratified them based on their lactate level at entry, and they followed these patients up to 30 days, and they had a look at who's surviving. So what they found was, if you had a lactate level of less than 2, which is normal, your survival was actually reasonably well. So at the 30-day mark, still about 80% of people are survived. But if you had a lactate level of higher than 10 at entry, your survival was pretty poor, and by 30 days, these patients had survival of about 30%, very poor. And that's because lactate is a marker of poor tissue perfusion. You're not getting oxygen. You can only rely on anaerobic respiration, which produces lactate. So that's the first part of our talk, and I hope it makes sense that oxygen is absolutely fundamental for cellular functioning by virtue of aerobic respiration to generate sufficient quantities of ATP. Thank you very much, and